Moving right along, let's talk muscle attachments, because before we can really talk about muscles, neuromuscular, anything, we need to talk about how the muscle interacts with the body, how the muscle interfaces with our skeleton. So essentially, our skeletal muscles are going to be attached to bones via something called a tendon. So you're going to actually run the muscle belly, the muscle fibers into a tendon. That tendon is going to be what actually connects the muscle to the bone. If we didn't have muscle attached to bone, we would not be able to translate our forces generated from our muscle into a mechanical movement, right? We'd still be able to have a level of mechanical work occurring because we'd be able to have a contraction of the muscle depending on what it was affixed to. But by having it placed above and below, or at an origin and an insertion, or proximal and distal, we're actually able to translate that force into displacement. So we're able to actually move a joint about and if we look at the origin and we look at the insertion, if we were to allow a level of mobility at the origin site and we had a contraction of the biceps brachii, which contraction just means shortening, what would happen? You would actually translate the humerus up and around. So you'd actually rotate the humerus in the glenohumeral joint because you would be shortening this level. You'd actually be pulling the arm up because you would be shortening across here, which that kind of defeats the purpose if we want elbow flexion, which is the prime movement action, prime joint action that's exerted by the biceps brachii is elbow flexion, right? So if we are able to largely immobilize the origin site and allow for a shortening of the muscle, we'll actually contract concentrically this muscle and allow for a change in joint angle. So by minimizing the movement at the origin, we can actually translate the force onto the desired bone, onto the desired bonus process or insertion point, however you want to put it, and allow for that translation of movement. Movements that decrease joint angle are going to be called flexors, and movements that increase joint angle are going to be considered extensors, right? So biceps brachii is going to be functioning as a flexor in this situation because if you have a normal joint angle of about 180 degrees roughly, and you decrease that joint angle and take that joint to 90 degrees, such as what it is here, you're going to be decreasing that joint angle, hence you're going to be flexing the muscle. Or going through a flexor movement. Extensors are going to be the opposite. If you extend that joint angle, if you go into a process of extension or lengthening or increasing that joint angle, you're going to be using extensors. What's the overall structure of skeletal muscle and why does it matter? Essentially, the muscle is going to be divided into a couple different sections or really subdivided down based on specific layers of connective tissue. So the outermost layer is going to be called the epimysium, and that's really what is going to wrap the entirety of the muscle. The perimysium is going to be what surrounds individual bundles of muscle fibers, creating fascicles. And then the endomysium is actually going to be what surrounds or wraps around each muscle fiber. So think about this almost like saran wrap, right? This is creating your fascia. This is creating your fascial borders, and it literally is like wrapping the muscle in saran wrap. And what you can see is that up here you've got the epimysium. If we go based on nomenclature, epi means outer. So epimysium is the outer. Perimysium is going to be right here. You can see actually that circle is highlighting the fascicle. The perimysium is going to be the like tendinous connective tissue almost that is running along the outside of that. And then the endomysium is all the way down here on the individual muscle fiber, wrapping that in. So it's kind of zooming in as you go through. When we go to talk about muscle cell structure, what I want you guys to think is that realistically, muscle cells are not dramatically different than other types of cells. They tend to have very similar features, very similar organelles, right? Like they have a cell membrane, they have mitochondria, they have lysosomes, they have these very specific things. The major difference is going to be that skeletal muscle cells and muscle cells really are going to be multinucleated. And what that means is that rather than one cell having one nucleus, one muscle cell can have tons of nuclei in it. And there's a very specific reason for that. And we'll get into that later on in this slide specifically. But essentially, The other major hallmark difference is that muscle cells have contractile proteins located within them. These contractile proteins serve to actually shorten the length of the muscle and pull each other over top of one another so that we can get a shortening of a distance. And it's these specific contractile proteins that give a muscle structure its shape, or its appearance under microscopy, or its appearance in histology, which is what you have over here on the right-hand side. And it's what gives the muscle fiber a striated look. Another aspect of skeletal muscle structure that you really need to pay attention to is going to be the presence of something called a satellite cell. And what a satellite cell is, it's a stem-like cell, or it's a dedifferentiated cell. And what that means is it can kind of technically go into whatever it wants. It can turn into whatever type of cell that it wants, or whatever type of organelle that it wants. But what these satellite cells do is they sit right underneath the basal lamina and right between the basal lamina and the sarcolemma, or the muscle, like the muscle membrane, if you will. And when those satellite cells get activated, whenever our body calls on those satellite cells, they actually move into the muscle cell to create more nuclei. So if you remember a couple seconds ago, I said that skeletal muscle is multinucleated. The reason that it's multinucleated is because of these very specific dedifferentiated stem-like cells that are called satellite cells. And as we continue to exercise, as we continue to stress our musculature, we actually increase the activation of these satellite cells and get these cells to move into the skeletal muscle and increase nuclei. Well, we don't really know specifically why we need to drive nuclei in. We know that it's because skeletal muscle fibers can be long, they can be big. And in order to maintain a certain level of myonuclear domain, we need to have more nuclei. And what that means is that myonuclear domain is essentially like the ratio of nucleus to myocyte, to muscle cell, and really think cytoplasm, if you will. So if we don't have enough nuclei inside of the cell, it's going to become dysregulated fairly fast because you're not going to have enough genetic material to tell the cell what to do. It'd be like one general trying to lead an entire army without having any subordinates underneath him that are able to exert control as well. It's just not going to function. It's not going to happen. The primary mechanical proteins or contractile proteins that we're going to talk about in this class are going to be actin and myosin. Actin is going to be your thin filament that's got a couple of different things to it. So actin is going to be your thin filament, and it's also going to have tropomyosin and troponin located on the actin. Myosin is going to be our thick filament, and our myosin is really going to be functioning like creating the function of Velcro, if you will. It's got like a hook and loop almost approach. It's got a binding head that will bind to the actin and facilitate the movement. Another really important aspect of the sarcomere or of the muscle cell is this concept of a Z disc, right? And you can see these Z discs here, and they kind of look like a ladder, if you will, or they kind of look like a vine going up and down. What Z discs do is they anchor actin and they anchor the myosin. So by anchoring actin to the Z discs and anchoring myosin to the Z discs, it allows us to pull each other over the other, pull actin over the top of myosin and contract. So we're actually able to shorten the distance between Z discs. So technically a concentric contraction would occur when we have a shortening of the distance between this Z disc and this Z disc. Continuing on with skeletal muscle cell structure, something else that we need to know about is the sarcoplasmic reticulum. And these are going to be membranous channels that surround the myofibril, so surrounding the muscle fiber. And that's what's going to be highlighted here in this kind of light blue. This is a storage site for calcium. I want you to think about this like a big grocery bag full of calcium. It's going to be surrounding the outside of the muscle fiber and just storing calcium. The transverse tubules are what's going to be highlighted in yellow. So you see how the SR really wraps around these myofibrils, whereas the T tubules travel deep into the muscle and they actually go all the way through and cut transversely, hence transverse tubule. These are going to be responsible for carrying the signal from the nervous system, or really interpreting the signal through the muscle from the nervous system and facilitating a later reaction. So sarcoplasmic reticulum, membranous sacs full of calcium, T tubules, they extend transversely across the muscle and they pass through sections of the muscle, deep in the muscle. The neuromuscular junction is going to be the site where we have a neuron meeting with a muscle cell, or meeting with a muscle in general. So the neuromuscular junction is simply the meeting site of a presynaptic neuron, a motor neuron, and the muscle itself. What happens in the neuromuscular junction is you have a signal that gets propagated down the axon and towards the terminal branches of the neuron, right? Of the motor neuron. You have synaptic vesicles that are filled with neurotransmitters. They can be various things. Oftentimes you see it referred to as acetylcholine, ACH. It really depends on specifically the type of innervation that's going on. But just know that you have synaptic vesicles full of neurotransmitters. When that action potential, when that depolarization makes it all the way down to the bulb, all the way down to the bottom of the neuron, the bottom of the axon, you're going to have this translocation of these vesicles. These vesicles are going to actually fuse with the outside of the axon at the very end of the axon, and then release their neurotransmitter into this gap. This gap is called the synaptic cleft, and this gap is the only space that exists between the neuron and the muscle. So these two things, the neuron and the muscle, do not touch. They do not interface directly. Rather, there's a neurotransmitter expulsion that connects the two, right? When you get a neurotransmitter, when you get a certain level of neurotransmitter binding on the muscle, you're going to cause a depolarization of the muscle. In this section right here of the muscle, this on the where you're receiving input from the neuron is going to be called the motor end plate. So I know I zoomed through that. There is a ton of information that goes a lot further in depth in both of the textbooks for 4460 or for 4450. But in general, what I want you guys to know, and what I want you to be able to say off the top of your head immediately, is what is the function of the T-tubule? What is the function of the sarcoplasmic reticulum? What is the general structure of a muscle cell? What does actin have that myosin doesn't? What's the function of myosin? Because that's going to help you as we're translating this muscle signal, this neuromuscular signal from the brain to the motor unit and causing a contraction. Sound good? Awesome.